of a second messenger being produced in the cell. And that second messenger then is the signal for the cell to do something. And that often works by turning on or turning off enzymes inside of that cell. So they're going to catalyze certain reactions or not. Okay? And those cell responses, again, you know, can be a multitude of things depending upon the system we're talking about. And that's what we'll see throughout the term are some of those specific examples. Right? We're just being general here. You get a cell response. What is the cell response? It depends, right? It depends on, it could be cell division, it could be secretion of something, it could be movement. It really just depends on what we're trying to talk about. Okay. So we'll look at that. The other thing that and I mentioned already, these hydrophobic things often affect your DNA expression, right? So a signal that can actually get into the nucleus, what's in the nucleus, your DNA, right, is going to affect the expression of that DNA. So they likely can serve as what we call transcription promoters or something like that, where they promote transcription. If you get transcription, then you get translation, you get a new product in the cell or something like that. This is very elaborate, actually. It's kind of cool the way some of those things work on a genetic level. And then lastly, again, because of their chemistry, a cell can produce a lot of hydrophilic messengers and store them. It can store them inside the cell in a vesicle, a membrane-bound vesicle. And they'll stay there if it puts them in there because they can't fit through the cell membrane, right? They're not soluble in the cell membrane. So cells will often store hydrophilic signals so that they're ready to release rapidly, right, when you need them. You've got a large supply of them to dump out when you need them. So these responses are often a little bit quicker in terms of releasing the signal. Hydrophobic signals, on the other hand, because of their chemistry, there's really no way to store them. If you were to try to put them in a vesicle when you make them, there are some that you can, but if you were to try to put them in a vesicle when you make them inside of a cell to store them, the vesicle's a phospholipid bilayer. What's it going to do? Leak right out, right? It's just going to diffuse out because it's, by simple diffusion, because it's soluble in that membrane. So for steroid-based hormones and signals, which they're all based upon cholesterol, you usually have to synthesize those on demand. So when the cell needs to release them, it has to just start making them and release them. And where this would occur, where it would produce them, right, it's not gene expression because we're not making a protein. It's taking a lipid that you already have inside of a cell, like cholesterol, and turning it into a signal. And that usually happens in smooth ER. Smooth ER is the organelle that does a lot of lipid biosynthesis. And then once it produces it, it leaks out and goes out into the cytoplasm and then out into whatever the extracellular fluid is, where it will be carried back. Does that kind of make sense? So keep this in mind. If you can understand these kind of things here, it makes the understanding of chemical signals a little bit more logical, right? If we can understand their chemistry, why they do the things they do makes sense, right? Because of their chemistry. Can't go inside, so we have to have this happen. Can go inside, so we have to have this happen. And that's, again, kind of related to it. Does that seem okay, just in a general sense? So let's put this now in context with the endocrine system and look at some, not specific examples, but specific examples here just focusing on hormones, but you'll see it's the same stuff we've talked about before. Some hormones are going to be what? Hydrophilic, and we'll have to have this discussion. Some hormones are going to be hydrophobic, and we have to have this discussion. Most of them are hydrophilic. So it's kind of just going to be a